Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and good day to all. So this is a recorded video for our previous lecture. Yeah. Firstly, I would like to apologize for taking some time to prepare this recorded lecture. I've been quite busy for the past 1 or 2 weeks, and this is the only time I have to prepare a recorded lecture. So we are going to look into the growth of microorganism and control. Yeah. For this on this lecture, we are going to look into the growth of microorganism and control. We are going to learn what are the factors that affect the growth of the microorganism and how we can utilize the knowledge that we have to control its growth. Yeah. So these are the outline of today's lecture. We are going to learn about basic, just a quick overview about food. We are going to learn about the microbial growth. So normally the microbial growth, there are two different ways that we can refer to the microbial growth. Firstly, is the cell growth, which is individual, which refer to the individual cell growth. Yeah, and then another term is the population growth. Yeah, this is the common way that we can refer to the growth of the microbes. And then we are going to look into the requirement for the microbial growth. Yeah, and focusing more on the factor that influence the microbial growth. So there are actually two different factors, yes, two different factors that affect the microbial growth. Firstly is extrinsic factor, and secondly is the intrinsic factor. So we are going to look what are the intrinsic factors that affect the growth of the microbe and what are the intrinsic factors. And then from the knowledge that we have, uh, uh, how we can apply the knowledge to control the growth of the microorganism. So this is a quick overview about food. You all know what food is, but we normally can separate it or divide it into several category. The basic aspect of food is normally food are rich in nutrient. They are nutrient rich. Yeah. Of food that we normally see is that there are food that can spoil quickly, and there are also food that can polish. Why is that happening? So actually, it's mostly referred to the condition or the characteristic of the food itself. So, and then from this characteristic, we will reflect to the extrinsic and also intrinsic factor that affect the growth of the microbe. So there are example of food that spoil quickly and moist food, neutral food, unrefrigerated food, or sliced meat, and food that can be spoilish or that take a longer time to spoil are normally the dry food, acidic food, refrigerated food, and whole meat. And we're going to see why whole meat can take a longer time to spoil compared to the ground meat later. Okay. So we move on into the microbial growth. What is microbial growth? Normally, microbial growth you can refer to two terms: cell growth and also the population of microbial growth. So cell growth always refer to individual growth, how the one cells can be divided into the daughter cell or the second or the third cells. And then what are the requirements that needs to be met in order to allow the cell to grow or to divide. Yeah. And then another term is population of microbial growth, which refers to the number of cell, not the size of the cell. When we talk about population of microbial growth, the most important term, the most important criteria is the number of cell, not the size of the cell. Obviously, if let's say we refer to the cell growth, the size of the cell will also take into account. Okay, these are the common, and in order for cell to grow, there are there are different ways for the cell to grow. Yeah, firstly, it's normally through binary fission. There are cells that can grow through budding and fragmentation, and in order for cell to grow, there are certain requirements that has to be met. So we will see there are physical, chemical, and also other type of other requirements as well, such as water, mineral elements, and also oxygen. Okay, normally, in this lecture, we are going to focus more on the prokaryotic cell cycle or cell growth, yeah, which is very common to the microbes that cause spoilage of food, yeah. So it's a process where the cell produces from a parent cell into a daughter cell, from the mother cell into the daughter cell. So as I mentioned earlier, there are different ways for it to divide, yeah. But the most common way is through the binary fission. So these are the process of how the cell can divide or replicate through the binary fission. So this is an example of a Bacillus lecheniformis, yeah, which is taken live under the microscope actually. So this is a live image, yeah. So firstly, normally the process will start with elongation of the cell and a replication of the DNA. Yeah. So and then after the replication, both of the DNA will move to the different directions. Yeah. And then this will allow the cell membrane and plasma to divide to form a septum. This is what we call as a septum. And then there will be a cross wall form. The cross wall will form completely and divide the DNA, and then the cell begin to separate to form a to daughter cell. And the characteristic of the mother cell will be exactly similar to the daughter cell, unless if there is uh, any mutation happen to the cell. So I have included the link for a YouTube video. Maybe you can also check from the YouTube video. Okay, so budding normally happen in yeast, yeah, where from the daughter cell there will be a small bud happen, and then the the yeast begin to develop the nucleus and also start to bud into a new daughter cell. And then this is budding in Hydra. Yeah. Okay, this is the process. Binary fission is from a single parent, doubles its DNA, then divide into two daughter cells. Yeah, usually occurs in bacteria. So most of the prokaryotic cells use this technique, which is a binary fission. And then budding, a small growth on the surface of parent breakout, resulting in formation of two individual. So we have seen this now. And then fragmentations. So some organisms can break into two or more fragments that develop into new individuals. So the fragment itself will develop into a new individual. Okay. Okay, so these are the common growth cycle in a batch culture or in a closed system of prokaryotic cell. Yeah. So normally when the bacteria, yeah, when the prokaryotic cells met all the necessary condition, met all the requirement, uh, met all the physical, chemical, and also other requirement that allows for the cell to grow. So normally we'll have this type of curve that represent the population growth or the growth of the microbes. Yeah. So this is we call it as a batch. We can also we can also refer it as a batch culture if, let's say, we inoculate the cell into a medium, yeah, into a non-medium, and then we cultivate it over time. So we will expect to see the similar growth, the similar growth curve as well. This is also true if, let's say, there is there's a bacteria that contaminate our food. Yeah. Let's say, for example, our mom cook chicken curry, like I mentioned before. Yeah. And then there's a contamination happen in the cell. So normally the growth of the microorganism will also follow the similar pattern. So the common pattern is a lag phase. They will have a lag phase, yeah, where at log phase, where lag phase is normally there will be little or no growth during this phase, yeah. And then there will be a log phase where an exponential growth happen, where a rapid growth of the cell happen during this stage. And then there will be a stationary phase and also a death phase, yeah. What is actually happening during log phase, stationary, and death phase? I will explain each of the phases, yeah. So during the lag phase, normally is an adaptation phase for the cell or for the microbes. So at this phase, normally the cell start to adapt with a new environment, and then it will take some time for the cell to adapt, and then at this moment, at this phase, normally there will be little or no growth. Yeah, this is what we call as adaptation phase. And then once the cell adapted to the environment, to the nutrient and environment of the certain environment, the environment of the condition of the environments, so the cell will start to exponentially divide, yeah, or will rapidly divide. At this phase, normally there will be an excess of nutrient and also space during at the, let's say, at the chicken curry. Yeah? So the chicken curry will also have an excess nutrient and also space for the cell to divide during this phase. So at this phase, there will be there will it will since the nutrient are enough, the space are enough, yeah. So the cell will divide happily, will grow happily during this stage. Uh, and the division of the cell will happen at the most optimal conditions. Okay, but however, after some times, when the number of cells in the chicken curry has been so much, it has been too saturated or too loaded, yeah, the cell will begin to enter a stationary phase. So basically, in the stationary phase, most of the nutrient depleted. Yeah. So at first there are many nutrient in the chicken curry, but since the nutrient has been used for the cell to divide to produce new cells, so majority of the nutrient has been depleted, and also there are also the space of at that time is also become very congested. Yeah, so there are so many sounds that begin to at that stage, and some sound they start to release some secondary metabolites that will sometimes inhibit the growth of other microorganisms. So at this phase, why it became plateau here is because the number of the new cells and also the number of the death cells is at a similar rates. So that's why it became plateau here. But after some time, so the number of cell death exceed the number of cell growth. So maybe at this time the nutrient completely diminish. So the cell will enter a death phase. So the number of cell will start to decrease. Okay, this is the explanation of for each phases. You may look into it later. Yeah, and then so this is the importance of exponential growth. Yeah. So there are in for each phases. Yeah. For each of the phases, normally during this log phase, the cell will produce mostly primary metabolites. Primary metabolites are the metabolites that are usually required for the production of new cells. But on the secondary, and there are some important product can be produced by cell at this stage. But there are at the secondary stage, it's a stationary phase. Normally at this stage, there will be no new the growth of cell becomes stunted, yeah, and there will be a lot of secondary metabolite being released by the cell. Either it is a extracellular metabolite or intracellular metabolites. So if let's say you are interested in producing some product from the microbe, you may model it, or you may first identify either the product is a primary or the secondary metabolites, and then from there you can start. Mm, you can design your experiment so that you can produce the desired metabolite according to the different phases. Yeah. So for example, these are some of the metabolite that are important that are produced during the exponential phase. So at this phase, some microbe can produce antibiotic resistant activity. It is very important for the plasmid production. Yeah. enzyme production, starting culture, and it is also used for the starting culture of the fermentation. So if let's say you are interested in producing culture in a fermentation process, so your seed culture should start from, you should be taken from the exponential phase. And then it is also good for the to be used as a culture, to be used as a dough in for bread production. We call it as a starter culture. At this phase, the most of the microbe is in the active form, in active growing form. So the time taken for the dough to raise will be faster. Okay. So for the microbial growth, this is when, if let's say if you want to grow it in the lab system lah, yeah. For example, if you want to grow the microbe into a batch lab system, so we can call, there are different ways. Firstly, we can use a batch culture, and also we can also use a continuous culture. These are different ways on how the microbe can be grown, especially in lab. Yeah. But if let's say the microbe that contaminate food, normally the concept is almost similar to the batch system, not the continuous system. So for a continuous system, we need to modulate the process, where we need to pump in fresh medium simultaneously with the pump out of the medium and cell simultaneously. This is if let's say you are interested in producing certain type of product from the microbes. But if let's say in the common spoilage of the microbe that cause spoilage in a common system, that in a so common food system where there is a contamination of bacteria, and the bacteria start to cause spoilage. So the concept will be almost similar to the batch culture, and it will have these typical growth phases, where it has lag phase, log phase, stationary, and also the dead phase. Okay, so for microorganism growth, there are some terms that are important, considered important. Firstly is the growth rate, where the change, where it reflects the change in cell number or mass per unit time. Yeah, and then there are also a term called generation growth. Yeah, where the interval of formation from two cell, uh, from formation of two cell from one cell. And then generation time, yeah, it is actually a time required for the cell population to double, or the other term that we can use is a doubling time. Because this is an example of generation time, yeah, where the time required for cell to double. So this is an example of one generation, where uh, one cell divided into two daughter cells, and the time taken for one generation to produce is known as generation times. Okay, so this is how you are going, you can identify, or you can calculate the number of the microbial growth. So if we consider a bacterium growing under ideal condition, in which the number of cells exactly double in every generation, the population size after a known number of generation can be calculated. So we can easily calculate the number of the generation after, if let's say we know what are the initial number of population is. So when we know the number of initial population size, we normally term as n naught. Yeah. So after one generation, how we are going to calculate n1 can be calculated by 2 multiplied by the n. For example, the n naught is 10. Yeah. And then 2 multiplied by 10 would be uh, 20. So same applies to the rest. Nah? Yeah. So this is after n generation. You can also calculate it if, let's say, after 30th generation. So you can calculate using this formula. So there's a formula to use that. And then obviously, that's why if, let's say, we notice some microbes when it's spawn the food, some microbe can spawn the food very quickly, and some microbe it may needs a longer time uh, to spoil the food. Or let's say, if let's say you eat any food that are contaminating any food that are contaminated with bacteria. Some cases, the symptom that appears, the food poisoning symptom that appears may take longer and some may take shorter. This is mostly reflected to the generation time required for each bacteria to grow. So let's say if your food is contaminated with E. coli, obviously the food can be contaminated very fast, yeah? And the time taken for you to get the symptom will be fast because the time required for one generation is only 17 minutes. But let's say if your food is contaminated with Treponema pallidum, yeah, which is normally contaminating certain type of food, yeah. So obviously it may require a longer time for the symptom to appear, yeah. It may require a longer time for this food to spawn. So and then these are the common medium. If let's say if you want to grow it in the lab scale, yeah, in the lab scale in the shake flask that I mentioned that I shown earlier. So these are the common medium used for the growing up to grow the sound. So obviously this is the time taken influenced by the medium used. Let's say these are we consider as the optimal medium. So 17 minutes is the generation time required for E. coli under ideal growing conditions. So if let's say the condition is not as ideal as the medium require that the growing medium doesn't really have sufficient nutrient, obviously it will take a longer time as well. Okay, there are ways for us to measure the bacterial growth. So we can use direct method and also indirect method. So for direct method, we can use microscopy and also viable cell count. Yeah, I believe some of you guys have done this plate and spread plate and pour plate. Yeah, and then after we incubate it, after some time, then we can then we can observe. We can calculate how many number of new colonies form. Yeah, and then there's another way where we call it indirect method where we check the turbidity. Yeah, where we can reflect to the density of cell. Yeah, culture density, and also we can also we can also measure through the dry weight. Okay, so for the direct method, normally for the direct method, yeah, the number of cells in the original sample will be too much. Yeah, too much. If we directly take the original sample and we directly do the spread plate or pour plate, we will not be able to identify or measure the number of bacterial growth. So how are we going to solve the issue is through the preparation of, we should prepare a serial dilution where we dilute 1/10 of the medium, of the original medium into a new tubes. Yeah, so 1/10 of this will be included into this, and then this in here it will be diluted 100 times. So let's say this is from the original culture, we take 1 ml and we insert it into 9 ml of the broth. So actually we dilute it 10 times here. And then from that 10 times, we dilute another 10 times, in which it is already 100 times diluted compared to the original strain. And same condition applies to the other dilution methods. So for bacteria, normally we always take around 10 to the power of 3, 4 to 6, yeah, depending on the how severe the contamination of the microbes. So for food sample, this is a good range for us to take, normally from 10 to the power of -3 up to -4. So this is normally the range that we normally take to detect that or to measure the number of microbial growth. Yeah, so And then from here we can calculate how many, we can measure the number of cells or colonies. Yeah. So this is another way to detect the microbial, to measure the microbial growth by using a microscopic count. Yeah. We can, this is a hymocytometer where you can calculate the number of cells within this grid, and then from the number of cells that we can calculate that we calculated into this grid. Sorry. Oops. Okay, and then we can calculate how many cells are there. Yeah, but however, if let's say we didn't apply some staining, so we cannot differentiate whether the viable cell and also the non-viable cell. So in order to detect which cell are viable, which cell are not viable, so we need to apply some staining method. Okay. So this is a way to check the viable cell growth, yeah, through spread plate and also the pour plate. Normally, we use PCA, yeah, plate count agar, yeah, to detect the number of microbe in growth. Then we only use 0.1 ml or 100 microliter to spread onto this agar, and then after incubation, we can start to detect the surface colony. So for spread plate method is we normally we can detect the microbe that can only grow on the surface of the agar, or we can call it aerobic microbe. Yeah. But for for the pour plate method, we can also identify the microbe that are able to grow aerobically and also facultative anaerobes. Yeah. So the way of how do we do it is normally we pipette 1 ml. So the volume is normally higher than the spread plate method, and then we will mix with the molten agar. So this molten agar will be around 45 degree, 40 to 45 degree. And then after incubation, we'll be able to measure the surface colony and also the subsurface colony. So from here, we can identify which microbe, how many microbe are the aerobic and also the facultative anaerobic microbe. Yeah. Okay, this is an example of the plate. So if without a proper dilution, you will have this much amount of colonies and you will not be able to measure. So we need to make a proper dilutions. Okay. So this is another way on how to detect the measurement of the microbial growth through the stability of the cell. Yeah. So we can use spectrophotometer. Yeah, we need to know how many nanometer suitable for detecting of the cell. So normally the common spectrum used for detecting the microorganisms is at a 600 nanometer. So the blood will pass through the culture. So we'll put our culture in cuvette, and then there will be the ability of the cell will be measured. So from the measurement uh, we can detect or we can measure the number of cell growth. Okay, so these are the example of method or techniques that we can use to measure the microbial growth. Now, okay, now we want to see what are the factors that influence the microbial growth on food products. So I've mentioned earlier there are two factors, yeah, extrinsic factor. Extrinsic factor are the factors that you can also term known as external factor that affect the growth of the microbe, and intrinsic factor are the factor that inherent to the food itself. Yeah, what are the characteristic of the cell, the pH, not the cell, the food. Yeah, and the characteristic of the food itself. Some food are sour, some food are natural, some food are, alkaline. So these are actually the characteristic or factors that inherent to the food. So some food are dry. So dry food normally has less moisture. So normally can less microbe will be able to grow on the cell or on the food. Okay. So this is the example of intrinsic factor, yeah, water activity, pH value, and also redox potential. So this is the factors that inherent to the food itself. Yeah. And then extrinsic factor are the environmental factor, yeah, such as temperature, relative humidity, modified atmosphere, packaging, and etc. So we can also modify the environment through technological processing and also contamination. Contamination can also affect the environment uh, of the food. Yeah. Okay, both of these factors will actually determine the behavior of the microorganism. Will determine the growth, the death, or the survival rate, and also the interaction between the microorganism. So if the growth, if the conditions are favorable for the growth of the cell, so obviously the growth will be uh, optimal. Yeah. But if let's say the growth are not in the optimal, the intrinsic and also extrinsic factor are not at optimal condition, so it will obviously affect the growth, the death, survival, and also the interaction between the microorganism. So, this is the intrinsic factor. Is that this factor inherent in food that include the pH, water activity, oxidation reduction potential, nutrient content, the microbial content, and also biological structure. Okay, and then extrinsic factor is like the external to the food that affect the microbial growth. Okay, we will first look into the intrinsic factor. Yeah. Example of intrinsic factor is the hydrogen ions version of pH, such as pH, moisture content, nutrient content in food, antimicrobial substance, and also biological content. So most bacteria can grow well in a neutral or weakly pH, alkaline pH, such as around 6.8 to 7.5. But some microbe can also grow at an extreme pH condition, such as mold can grow in between 1 to 1.5 to sorry, some bacteria, some microbe can only grow in a narrow pH condition range, such as some bacteria, such as Salmonella, it can only grow between 4.5 to 9 pH. Yeah, but some microbe can also grow in a broad or in a wide pH range. Yeah. For example, mold can grow between 1.5 to 11 pH, and also yeast can grow between 1.5 to 8.5. So this characteristic will affect, yeah, the ability of the microbe to grow on the foods. So these are the pH value of some food product. If you see, most of it are within the range, they're not within the within the pH that optimal for the growth of the microbe. That's why most of food can easily be contaminated by bacteria, yeah, or bacteria or microbes. So some microorganisms also they can we call it as extreme. They can grow it in extreme condition, or there are terms that we can use for this microbe. We can call it as extremophiles. Yeah, some microbe can grow at the acidophilic micro condition. We can call it as acidophilic microorganism. So, for example of acidophilic microorganism are yeast and mold, where it can grow under extreme acidic condition. Yeah, where Vibrio chlorella sensitive to acid and prefer alkaline condition. Okay, but most microbe can be killed with strong acid or strong alkaline environment, except for the mycobacteria. So there are different ways. So we cannot use pH control strategy to inhibit the growth of the mycobacteria. So we need to use another techniques. Okay, so these are the minimum and maximum pH for growth of some specific microorganism. So basically, normally bacteria is around 4 to 9 pH 4 to 9. Yeah, for most bacteria, but mold has wider ability to grow, wider pH availability. They can grow in a wider range of pH, yeast and mold. So yeast and mold can grow in a wider pH. That's why if let's say we prepare a medium for yeast and mold, we normally make it acidified. So we normally set the pH to 3, yeah, so that it will inhibit the growth of most bacteria. So we can call it as a selective agar as well. And then these are the factor. This moisture content is the second factor, intrinsic factor that can affect the microbial growth. Yeah. So obviously the water activity term as the amount of free water in the food medium. So and the amount of free water is very important for the growth of microorganisms. Yeah. So the pure of the water activity of a pure water is equal to 1, and most food product will always have a water activity of less than 1. Yeah, especially the dry food. Yeah, and a saturated salt solution have a water activity of 0.75. And we will see later on most bacteria they will require a higher moisture content as compared to the yeast and mold. So if let's say that's why for most of the dry food product, it can be hardly contaminated by the bacteria. So salting and drying reduce the water activity of the food products. So we can use salting and also drying procedures to extend the shelf life of the food product. Okay, so this is the water activity of some food products. So raw meat and milk will have the water activity from 0.99 to 1, yeah. And some dried grain will have the water activity of 0.8. So, and okay, so this obviously this is what we have discussed earlier. The growth of the microbe can be affected greatly by the water activity. So you can inhibit the growth of the microbe if we inhibit the water activity level, and we can also maximize the growth of the microorganism if, let's say, we make the food to be uh, has a higher water activity level. So that's why moist food will have a higher activity level, and it can be easily spoiled or contaminated easily by the microbes. And this table here shows the minimum of the activity that support the growth of some microorganism. Yeah. For example, Clostridium botulinum required 0.95 of the activity. Yeah. And most bacteria will require at least over 0.85 water activity. But for most yeast and mold, it can grow at the lower water activity. So that's why if let's say most dried product, if you notice, if let's say there's any contamination that happen, normally is mostly caused by the yeast and mold. And then the third factor is the nutrient content of the food. Yeah. So obviously, in order for the microbe to grow, it requires a lot of macro and also micronutrients. Yeah. So it requires protein, carbohydrate, and etc. in order for it to grow. So the number of nutrients. Okay. So because of this nutrient-rich content of the food, it can be easily susceptible to the microbial spoilage. Yeah. So okay, the fourth factors is antimicrobial substance. Some foods it has antimicrobial substance, yeah, where it can inhibit the growth of the certain microbes. For example, lecithin and anticoliform factors can be found in meat, and also lysozyme in egg. These are some of the antimicrobial substance that can be found in milk and also egg. There are many other antimicrobial substance, especially in spices. Yeah, in spices there are many more antimicrobial substance that can inhibit the growth of microorganism. Okay, and then the fifth one is the biological structure. Okay, so meat has fascia, so that's why this actually differs. This actually affect the growth of the microbe uh, where we in our earlier slide where we find that ground meat ground meat can get contaminated easily as compared to the whole meat. So this it is actually attributed to the uh, uh, this structure we call it aphysia, where skin and other membrane, yeah, meat, the fascia, skin and other membrane that prevent the microbial entry. So the whole meat has the fascia, skin and other membrane that will help to prevent the entry of a microorganism. But if let's say the ground meat will not be having some structure, the structure has already destroyed, so that it will be easily that meat can easily contaminated or spoiled by the microorganism. Okay, another example is egg have shell and inner membrane that prevent yolk and egg white from infection. So we have seen this in our previous lecture. And also some fruit that are in acidic condition, for example, oranges, lemons, yeah. So the pH will be at a very low pH, yeah, and this condition will severely affect or inhibit the growth of most bacteria, but still will allow the growth of the yeast and mold. Next is the extrinsic factor. These are the external factor that affect, yeah. For example, a temperature, presence of concentration of gases in the environment, and also relative humidity of the food storage environment. We'll see first about, so we go to the first factor, which is the temperature. So the growth of microorganisms is affected by the environmental temperature, and obviously most microbes can normally grow at the ambient temperature, or we call it as the room temperature. Yeah, but some microbes that we can term it as extremophiles can also grow at quite an extreme temperature. So the microbe that can grow quite at the extreme low temperature, we call it psychopaths. Yeah. So normally it can grow within the range of -15 to 15 degree. And then the optimal condition or the optimal temperature for the microbe to grow is normally around 10 to 45 normally. So around this temperature, where we call it as the room ambient temperature, and the optimum will be around 30 to 40 degree. Yeah, 25 to 40 degree. But so the microbes that are able to grow, or most microbes that are able to grow at the this range of temperature, we call it mesophile, and there are also extremophile that can go at the hot temperature, we call it as the thermophile. Yeah. Okay. And then so for the psychrophilic microorganism, it's able to grow past at 420 degree, but also can be grow down to -10 degree in unfrozen media. So normally that's why if that's why if let's say we keep our food in in fridge or in refrigerator. Normally the temperature is around 4 degrees Celsius. That's why you don't surprise that still, if you keep it over a longer period of time, the food can still be spoiled. Yeah. Uh, and then okay, this is the common range of temperature that can be found by most bacteria. Yeah, we should be within 25 to 40 degree. And the optimum temperature will be around 37 degree. Yeah, so and some microbes such as Pseudomonas aeruginosa may even have a wider range of growing temperature, which is between 5 to 45, 43 degree. But none of the mesophilic bacteria are able to grow below 5 or above 45 degree. So, and mind you that most of the pathogenic bacteria belong to this group. That's why it is very easy for you to get sick if you get contaminated with bacteria that are grown at let's say you keep your food um, at this range of temperature, ambient temperature, at the room temperature. Okay, next is thermophilic bacteria. These are the type of bacteria that are able to grow at temperature over 45 degree, and the optimum will be 50 to 70 degree. Yeah, but there will be not much bacteria that are able to grow at this temperature. And so bacteria in this group are mainly spore former and important in the food industry, especially in the processed foods. Okay. Okay, note that the effect of temperature on the microbial growth also depend upon the environmental condition as well. So obviously, the external factor will also need to take into consideration about on the intrinsic factor as well. Yeah. And then consideration of the gases in the environment, depending on the type of microbes. Some microbes require high oxygen tension, some microbes that require low, they are able to still grow in a low oxygen tension, and some really need absence of oxygen, we call it an aerobic. Yeah. But some microorganisms also either grow in either high oxygenation or absent of oxygen, such as facultative anaerobes. Okay. And then thirdly is actually the relative humidity. Yeah, relative humidity is the amount of moisture in the atmosphere or food environment. Food with lower water activity placed at the high humidity environment take up water, increase their water activity, and get spawn easily. So obviously the relative humidity of the environment also can affect the uh, ability of the microbe to spawn any food products. For example, dry grains stored in an environment with high humidity will take up the water and undergo the mold spoilage. Okay, so we have learned about the intrinsic and also the extrinsic factor that affect the microbial growth. So we can actually use this knowledge to develop to manipulate the growth of the microbe or to hinder the growth of the microorganism. We call it as the hurdle technology. Hurdle technology is a method to ensuring that pathogen in food can be eliminated or controlled. This means that the food product will be safe for consumption and their shelf life will be extended. Hurdle technology usually work by combining more than one approach. So this is an example of hurdle technology. So we can use different factors, yeah, to inhibit the growth of microorganism. Let's say we can combine two different intrinsic or extrinsic factors. Yeah, for example, we use pH preservation, where it will inhibit the growth of many microbes, but still some microbes can also tolerate high pH condition or extreme pH condition. But if let's say we combine it with controlling the water activity, so we will be able to control the growth of the microorganism, where the package will need no refrigeration hurdles. So it will also it will save the cost for the keeping, yeah, for the storage. And also we can also combine many more factors. If let's say it is in the food, let's say the food already has a very high water activity, where it can it can easily get spawned. So we can combine with many of the other factors to make sure that there will be no growth of the microorganism. So this image, I think, can clearly help you to understand the other technology. Yeah. All right, so this is all for this recorded video. I will upload this video in your portfolio, and I hope you understand and enjoy the video. Thank you.